A variety of different data, both population genetic data and ecological data, we can study with thousands of genotyped mangroves in the 4 major estuaries of Panama. But we're also going to combine that. All of the trees I'm talking about are actually pine willows are blooming, won't forget it. They produce these spectacular, lose all their leaves and produce these spectacular floral displays that you can see from space. And we also use a series of 5-6 hectare plots that you see in yellow. These are centered around 40-meter tall automated telemetry towers that have been used to do animal tracking across the island.
If they're failing to reach a lot of sites, then as soon as you drop out more sites, they're unable to produce enough seeds to reach enough sites to maintain themselves, and they drop out of the system. In contrast, if we go to the tolerance fecundity model, the critical question for extinction risk concerns the continuing availability of sufficient regeneration sites representing the full range of stress tolerance. Look at species traits. How does fecundity vary with seed mass? How does the stress tolerated vary with seed mass? And also the environment. So what's the distribution of sites with different habitat stressfulness? What's the distribution of sites with less than some level of stressfulness? Because it's these things together that will determine the relationship between
dynamics. If we discard metapopulation dynamics, extinction debt may not be such a problem. But there are many other issues, so we have to be very careful of our assumptions. And so in this case, as I say, there are many other types of regional dynamics. We've shown in our very particular system that probably don't have metapopulation dynamics, and we have other field evidence suggesting that as well. But many of your systems may not as well.
After our delicious lunch. Yeah, so I'm going to talk to you guys about tracking seeds using some strings. Oops. Okay. So I'm not going to go into a lot of detail about this diagram. The solid lines are survival events, the dashed lines are death events, usually through predation.
Typically we will use very polymorphic markers like microsatellites, so that in general it's information when you fit a dispersal kernel, if you have a very complex dispersal pattern. But you should report these frequency sites. Could be seed tracks or could be just seed collection sites, a kernel function directly. This would not be a kernel. I mean, if we can incorporate it in the model, and in the same way as we saw before for neighborhood models, let's
see how this works. So I'm going to talk to you about inverse modeling and all the seed sources in the neighborhood. And that's individual seed shadows. Obtaining some seed shadow and just continue iterating the models and fitting different models until the some seed shadow corresponds to what we see in the field. So well, that brings me to our case study where we applied inverse modeling. It was a collaborative study, most protected by hard endocarp and fruits. Thank you, Patrick. After our. So we have. It has a lot of quite large dispersal community, which include capuchin munchkins,
which is an endemic to the Society archipelago in French Polynesia. And in addition to that, there's the silver eye and the red-vented bulbul, both of which were introduced within the past 100 years. Tahiti and Moorea, unfortunately, were also the site of a particularly catastrophic story of invasive species. So Myconia calvescens is dispersed by birds and was introduced to Tahiti in 1937. And from its site of introduction, it spread across Tahiti and now is estimated to cover nearly 70% of the surface of the land. On the neighboring island of Moorea, it didn't arrive until around 1960, and currently only covers between 20 and 30% of the land. So the extent of invasion is much less dramatic on Moorea, how long it takes for seeds to be passed. So this diagram is based on fieldwork from 2008, in which I identified 26 species that are dispersed by 3 frugivores. And here I've represented native species in yellow and exotic species in blue. And you can see that exotic species are well integrated into native seed dispersal web.
These, which in theory can be introduced into an ecosystem which has lost a native species. And this non-native species will replace the ecosystem functions of that native species. Taxon substitutes in Mauritius, focusing on the island of Elizagrat. Now, Lisbon is a 26-hectare island, and the dominant vegetation is lowland coastal forest, and this species, Diospyros agroterum, which you can see here, foliing. Now, we can rule out these guys for dispersing ebony because they're actually too small, and we also know that a lot of them don't consume the fruit, except for the talcum skink, so it probably wouldn't have been an effective seed disperser because it could only consume very small seeds.